Hello, grade 12 students. In this video, we're going to talk about the exam 1 for grade 12 LS. So this the series of our biology exams for the academic year 2018-2020. So let's start first with question number 1. Question number 1 is a genetic question. They said that document 1, which is this document, represents the pedigree of a family with some of its members shown in black, these one, that are shown in black. You have a rare hereditary disease that occurs mainly in males and very rarely in females. From this sentence, you have to know that this disease is sex-linked disease. Is the allele, first part, is the allele responsible for the disease dominant or it's recessive? Justify your answer. So, first of all, you have to know if it's dominant or recessive, justify your answer. Let's have a look at the pedigree. Having a look at the pedigree, 1-1 and 1-2, they are normal. But they gave birth to 2-9, which is affected. So this affected person have normal parents. So the allele coding for the disease was masked in the parents. This is the most important word you have to say here. You have to say this key word. You have to say it's masked. So the allele coding for the disease was masked in the parent. So the disease, the allele of the disease is recessive with respect to the normal dominant allele. This is the first part. Did we finish part A? Sure not. We didn't finish part A. Why? Because we have to give symbols. So some of you in the exam, they only say which one is dominant and which one is recessive without mentioning without giving the symbols. So you have to give the symbols. So how come you're going to move to part 2 without solving, without giving these symbols? So you have to know that. And 1-1, as I said, 1-1 and 1-2, they are normal, but they gave birth to an affected child, which is 2-9. So the allele of the disease was masked in the parent. So it's recessive with respect to the normal dominant allele. Then we have to give symbols. Let and capital be the symbol of the normal dominant allele, which is dominant over the recessive allele of the disease. And let D small, A small, whatever. Give it a symbol that you want. Mostly it's a disease, so give it a D small. Be the symbol of the allele coding for the disease, which is recessive. For 2, they said, discuss logically the chromosomal localization of the gene responsible for the disease. They said without considering the female 20. Without considering female 20, meaning that female 20 is not in the consideration. We cancel this female 20. So if you see here, mostly the affected, they are males. So it's sex thing. We remove the case of autosomal. So autosomal case is here cancelled. You have a sex thing one, either in the X or in the Y or XY. If it's on the Y, every affected male should have an affected father. For example, 2-9 here is affected. So his father is normal. So it's not in the Y. So he inherits the Y from the father. So it's not the case. So the allele coding for the disease is located in the non-homologous region of X. It's found in the X chromosome. 43. Illustrate chromosomally the genotype of each of the individuals. 13 and 16, justify your answer. Illustrate, you have to draw these chromatids, for example, and give them the alleles. So, illustrate chromosomally the genotype of each individual. 13 and 16, justify your answer. So here we're working about sex chromosomes, X and Y. 13 and 16, you have to justify. Let's move to 13. 13 is a male. We said that the allele of the disease is found in the non-homologous region of X. So at 13, it's a male and it's affected, and the allele of the disease is recessive. So we have only in 13 we have XY. So we have X and we have a small y, and we have the allele of the disease. 13, it has only one allele, which is D. That's why it's affected. While 16, it's normal. This lady, 316 here, she's normal. So she had the normal allele, which is dominant. She has one of the normal alleles. Since it's female, it has 2 X alleles. So she's normal. But she gave birth to affected children, which are 23 and 24. So she's a carrier of the allele of the disease. So she carried the allele of the disease, but was masked in by the dominant allele, which is for the normal. So she'd have an D. Okay? Let's move now. Female 20 presents beside her disease an abnormality, which is manifested by the absence of menstruation absence of the development of the mammary glands and other problems. So the female 20 has a lot of problems. Besides her disease, she has a disease, she has other problem by the manifested by the absence of menstruation, absence or the abnormal development of the mammary glands. To identify this abnormality, we perform the karyotype of female 20. This is the karyotype of female 20. If you have a look, all of them they are normal from 1, 2, 3, 4, 5, 6, 7, 8 till 22. All of them they are normal. We have 2 copies of each chromosome. So we have the abnormality here in the sex chromosome. This female should have 2 X chromosomes, but here it has only 1 X. So the abnormality here in the sex chromosome having 1 X, what do we call it? The Turner syndrome. So she had the Turner syndrome problem. So the Turner syndrome is the abnormality here. So it has only 1 X. So the chromosomal formula and give the abnormality revealed by the karyotype. The chromosomal formula here, we have only 2n equal 46 in the normal case. Here we have 2n minus 1, since we have a missing chromosome. 2n minus 1 equal 45. It's equal to 44, comma, X, 0. Abnormality is called the Turner syndrome. Based on the karyotype, how can you explain the appearance of the disease in the female 20? The female 20, appearance of the disease had only 1 X. So she had no possibility to have another allele, for example, let's say N. So this reveals or explains the presence of this disease in the female. So question number 1, as I said, the allele responsible for disease is recessive, since individual 9 is disease, while his parents, couple 1 and 2, they are normal. So they received the allele of disease from his parent, which was masked. So important to say masked. Let N be the symbol of the normal allele, and let D small be the symbol of the allele of disease. Localization for B. Since the disease affects mainly the males and very rarely the female, this is something that I told you. This is very important to tell you the disease is sex-linked. So if the gene is located in the non-homologous region of Y, as I said, transmission should be from the father to the son. But 9 is affected, but his father is normal. So it's not the case. So it's found on the X. Or C, illustrated chromosomally. So we have chromosomes X and Y, and here female 16XX. So here 16 is normal, so should have the M chromosome, but she gave birth to affected children, so she'd have the allele of the disease, which is D. So here, chromosomes 2X. Here, we have X and Y. Some of you are going to ask me why we didn't put here another D, for example, because it's affected. Wait a minute. This disease is found on the X chromosome and not on the Y. So the Y shouldn't have any chromosome. Chromosomal formula, 44 plus X, Turner syndrome. And this karyotype shows only 1 X chromosome, having the sick allele, and this doesn't have an allele on the homologue on the other X, so the disease will be expressed in this female. Moving to question number 2. Question number 2 related to the neurophysiology, to the nervous system. The framework of studying some aspects of the control mechanism of the muscle activity during dances. So studies were carried out and summarized in the document 1. This document 1, the movements of a dancer are performed in sequences, which are not always predictable, since each of these movements are triggered by an intention. So they are followed by an intention. So this will explain that we have a voluntary motor activity. This voluntary motor activity, usually it's the one responsible, or the nerve center responsible for this, is a part of the brain, which is called the cerebrum. The body is then used as mean of expression. However, any body's movement is hindered by a force, which is called the gravity, which attracts it toward the ground. To control the body movement and to reach equilibrium, the dancer uses muscles that block some joint movements and prevent their falling down. The posture is thus maintained thanks to a constant adjustment of the muscle. For instance, every time a muscle is stretched, it contracts. So every time a muscle is stretched, it contracts. This sentence explains that we have a motor activity, voluntary motor activity, or we have a myotatic reflex. So we have a myotatic reflex. So myotatic reflex, muscle stretched, as muscle contract as it's stretched. So this explains that we have a myotatic reflex. Part number 1, they said that pick up from document 1 a statement that justifies the presence of a myotatic reflex, and another one that justifies the presence of the voluntary motor activity. So myotatic reflex a muscle stretched and contract, as a muscle stretch contract, and the voluntary motor activity, the movement is triggered by intention, or you can say it's followed by intention. Specify the nerve center responsible for each of these activities. Already I said that the myotic reflex is the spinal cord, while the voluntary motor activity it's the voluntary motor activity it's the cerebrum, the part of the brain. In order to understand the functioning of the neuron circuit implied in maintaining posture during dancing, and to know how the muscles interfere in maintaining the body's equilibrium, the following experiments are performed. First experiment, a nerve fiber issued from a neuromuscular spindle of an extensor. Because the extensor is the triceps ray is stimulated. Triceps ray is found in zero. Triceps ray, and we have the anterior tibialis or tibialis anterior. This stimulation leads to the modification of the electric status of the two motor neurons, M1 and M2, located at the level of the gray substance of the spinal cord. One of these motor neurons innervates the extensor, while the other innervates the flexor, the anterior tibialis. So extensor, triceps, flexor, anterior tibialis, or tibialis anterior. Document 2 reveals the experimental setup and the results of the recording. Before we start here, let's see here, whenever we have excitation at level E1, we have decrease in the negativity, which is called hypopolarization. This will lead to the appearance of an action potentials here at the level of E3, at the recording E3. So E1, we have at E3, we have action potentials. So we have an action potential, so here the message is moving. While the level of E2, the recording at E2, we have the increase in the negativity, which is called hyperpolarization. So there is no recordings here. So we don't have recordings. Analyze the obtained results and draw out the effect of the activity of the motor neurons of the concerned muscles. So here we have contraction of the extensor and relaxation of the flexor. So here it relaxed. The second one at E2, which is the anterior tibialis, we have the relaxation since we don't have an action potential, while here we have contraction since the message reveals the presence or recorded the presence of many action potentials, APs. Referring to the recordings E1 and E2, determine the number of synapses implied in each of the concerned neuron circuit, that the transmission of the nerve message at the level of the synapse needs 0.5 milliseconds. They said that it needs 0.5 milliseconds, but the level of E1 needs about 0.5 milliseconds. So we know that it contains only one synapse. So since it needs only 0.5 seconds to move, so it contains only one synapse. So this is called a monosynaptic one. While in the second one, at the level of E2, it needs more than 0.5 seconds, approximately 0.7 to 0.75. So this explains that it contains more than one synapse. So this we call it the polysynaptic. So the question number 2, myotic reflex. Each time the muscle stretches, contracts, this sentence explains the presence of the myotic reflex. Number 2, voluntary motor activity. Certain movements, they follow an intention or they proceed by intention. Every one of them is 0.25, so both of them, they are 0.5 marks. Number 2, myotic reflex. The center of the myotic reflex is the spinal cord, while the voluntary motor activity is the cerebrum, 0.5 marks. The stimulation of a nerve fiber issued from a neuromuscular spindle of the triceps ray allows the recording of hypopolarization. This one that I said, hypopolarization at the level of E1. Hypopolarization, we said that polarization is negativity. Hypo, decrease in the negative, so we're going to go to the positive. Okay, so the stimulation of the nerve fiber issued from the neuromuscular spindle of the triceps ray allowed the recording of hypopolarization. So this is important to say, hypopolarization at E1 based at the level of the motor neuron M1. This message is an excitatory one. Why did you say that this is an excitatory one? And you have to say that this is an excitatory. Why? Because this leads to the appearance of many action potential recorded at the level of E3 based at the efferent fiber of this, which is linked to the triceps ray, which is the extensor. Okay, so this will lead to the action potential, as we said here. So the excitation here, we have the recording at E1 leads to the presence of E3 of action potential at the level of triceps ray. So we have an excitation. So the message moves, it doesn't stop. If it stops, we say that this message is an inhibitory one. while it moves and shows action potential, meaning that it's an excitatory movement. While at the level of E2, stays at the level of the motor neuron M2, we have hyperpolarization. Hyperpolarization, if you have to remember this. Hyperpolarization, polarization negativity, hyper increase in the negativity. So this explains this case. This is the hyperpolarization. Okay, while in the first one at the level of E1, motor neuron M1, we have the hypopolarization. So hyperpolarization was obtained and no recording were obtained at E4. So there is no action potential, which is linked to the anterior tibialis. So this one, we have no action potential at the level of E4. E4, as you know here, is connected to the anterior tibialis, okay? So the flexor one. So since M1, it sent an excitatory, as we said, to the triceps ray. The triceps ray contracted. While since M2 does not or did not send any message to the anterior tibialis, the anterior tibialis remains relaxed. So here, the relaxation is for the flexor, which is the anterior tibialis. Therefore, the variable activity of these two motor neurons, they will lead either to the contraction of the extensor, to the contraction of the triceps ray, since we have an action potential, and lead to the relaxation of the anterior tibialis, which is the flexor, since we don't have any action potential. As I said, the nerve message transmitted by the afferent nerve fiber took approximately 0.7 milliseconds to reach the motor neuron M1, and the time needed to cross the synapse is 0.5. So one synapse exists along this pathway. So this circuit is monosynaptic. While the nerve message took about 1.2 milliseconds across M2. So this means that it contains more than one synapse. Mainly it contains two synapses. So this is called a polysynaptic circuit. Anterior tibialis, number 5. Let's move to number 5. Second experiment. We said experiment 1 is replaced at the same time we stimulate a nerve fiber issued from the superior nerve center related to the motor neuron M1 that's linked to the tibialis, anterior tibialis. Many action potentials were recorded at E4 and no recording was obtained at E3. How can you specify the effect of these muscles? Justify your answer. So here, anterior tibialis contracts and the other relax. What do you call these two muscles? Anterior tibialis and triceps ray. They are called antagonistic because they have opposite. When the one is contracted, the other relaxes. They are called antagonistic. So the message coming from the superior nerve center inhibited the message arriving to the triceps ray. It stopped the message to reach the triceps ray since we have no recording at E3. However, at the level of E4, which is located at the tibialis anterior, we have the message reached, so we have a contraction. So this explains that the anterior tibialis will contract and the triceps ray here will relax. So when one of them contract, the other relax, and the opposite is true.